So my name's Katy Graef, and I'm from Ventures for Global Health, which is a nonprofit based in Seattle, Washington. We are the partnership hub administrator of WIPO Research, which is a global consortium dedicated to accelerating product development for neglected tropical diseases, malaria, and tuberculosis. And I had a presentation prepared, but I know since we're short on time, I'll just go through this really quickly. So as I mentioned, it's a global consortium. We have over 100 members across the globe, and these are from 29 different countries across the 6 continents. This includes academic organizations, nonprofit organizations, government agencies, as well as pharmaceutical companies. And the goal is to facilitate collaborations between these two, or between groups within WIPO Research, to allow the sharing of intellectual property to advance product development. As they said, BVGH acts as the academic matchmaker of the consortium. So we're the group that examines different members' research, finds out what their needs are, finds out what their interests are, and then finds another partner within the consortium that's able to contribute the asset that's needed to advance that other researcher's efforts. So we were established in 2011, and since then we've facilitated over 100 collaborations. And importantly, these collaborations are global in nature. We have groups from Nigeria collaborating with groups in China. We have groups in Cameroon that are collaborating with groups in Canada. So it's a really important feature of the consortium that we are getting ideas from all across the world and advancing research all across the world. Most importantly, this research, the goal is to advance product development. And so we're starting to actually see some of the effects of these collaborations. So we have our pipeline here, and I just have 4 of the diseases we focus on. And you'll see that in some instances, we have a line going up from a black box, and that line represents movement along the product development pipeline. And so, as I said, WIPO Research is about sharing IP assets. So we have everything from compound libraries to data to reagents or just know-how and expertise. And I'll just go with one collaboration here so we can get on to the panel session. But I believe earlier today, capacity building was brought up as a really important aspect of research in NTDs. And I think one of my favorite collaborations is a collaboration between the Noguchi Memorial Institute for Medical Research in Ghana and the University of Washington in Seattle. And we put Regina from Noguchi together with Sue Kramer from the University of Washington, and they are actually co-leading a University of Washington undergraduate course looking at the role of SNPs in CYP genes of Ghanaian populations and seeing whether or not there are actual differences that might relate to different gene metabolism. So we have Sue's lab, who has the expertise in the bioinformatics. You have Regina, who comes with the idea and wanting to know what sort of drug metabolism might be different between different populations. And then you also have Sue Kramer's undergraduate students who are actively participating in this research. So just a couple other collaborations there, but just to give a framework for why I'm here is WIPO Research is a free access, open access platform for all nonprofit or non-for-profit entities. It fosters collaborations, and the end goal, again, is to accelerate product development for neglected tropical diseases. So with that, I will start the panel session. And so I think the first question I had was, you all have done such really great work in coming up with interesting platforms to facilitate and foster open innovation. And how can we get groups to actually use your platforms or expand on the platforms you have? So Tale, would you like to kick it off?
I think for us it's just initiating a collaboration on its own. It's starting working together. It's sort of the key, and then building trust amongst each other. And I think for us in the European Lead Factory, but also in other collaborative projects that we've seen, trust is sort of the key ingredient, and therefore you need to know each other. So I think it was earlier mentioned by somebody, you cannot start a collaboration with somebody you cannot even have dinner with. I think if you don't know a person, it's not going to work. So it's all about networking, as also been said. And from those networking discussions, I think come great ideas, and you have to pick those ideas up and see if there's any other people in the room that want to collaborate on this. And then also share the data. There needs to be trust to share your generated data and not just keep them by yourselves. I think that accounts for me.
Do you want to just maybe go down the line if you have anything to add to that?
I mean, I would support what's said. I think it's important to make sure that we aim for win-win collaborations where the community benefits, but also the individual researchers benefit, recognising their needs to perhaps have publications and work for students. So I think it is that community of trust. I think it's also recognising that sometimes our funding gaps and actually finding funding for people to do these small projects is challenging, and that's something that the community needs to think about, I think.
Yeah, I think one of the SGC's strengths in collaboration is that we don't always feel like we need to take the lead. I think by and large people know what our strengths are. They come to us sometimes, can you make this protein, can you crystallize this protein? And sometimes that's all we do, and then after that we let our partners take the data and do whatever they want with it.
Yeah, I echo the comments around some of the gaps that exist in the value chain, really. When we're speaking to universities, we often find that it's a bit of a chicken and egg situation. They've not developed a compound collection because they don't have the infrastructure to screen, and they don't develop the infrastructure to screen because they don't have a compound collection. We have within AstraZeneca, we have the capacity to give out far more sets of compounds in assay-ready format than we have the capability to screen them for partners. But we find that the limitation is that relatively few partners have actually got the infrastructure to do the screening. So the offer is often there, and it's just left hanging, which is a shame. And I think there's a funding gap. There's an infrastructure gap, and there's also a training gap as well in terms of the medicinal chemistry skills to make use of the output in the early research stages. So initiatives like the European Lead Factory, I'm certainly very keen on, because I see it as helping to address all of those issues.
Great. And so I think you all at one point in time mentioned that funding was kind of it's a very significant barrier. And Paul, I know that you have your Endemic Country Researcher Award that you had actually put out to solicit individuals to actually screen the pathogen box. And do you see kind of that as being a new model for getting people to use different open access platforms, as maybe, dangle a carrot of some funding, get people going, get the trust going? Or what do you guys think about how we can actually fund these open access initiatives more?
I mean, I think that was a gap that we were trying to fill, and when we talked to a lot of researchers in endemic countries who'd requested to take the malaria box and put it in the freezer, it was just because they didn't have the money to run the screens, so they were saving them till they had that money. So this is a way of actually trying to give them what are relatively small sums of money that they need to actually to do that work. So, I think that's something that works in endemic countries. There's also other initiatives that have worked in other countries. So, for example, there were Gates Grand Challenge grants that also were launched around the malaria box that allowed researchers to do work there. And we're hoping that something might happen there in the pathogen box area next year.
Excellent. And do you think there's ever a time when too much open access is, is there such a thing as too much open access? So Raymond, you talked about how you make sure that before the data is shared with the public that it's been reviewed and it's really high quality. So is there a situation with everyone where open access is not the solution?
I think that's too difficult to answer in a general sense, but it wasn't that long ago, I think there was a publication in Nature talking about how a significant percentage of research data simply cannot be reproduced. And I think that's of grave concern to not just infectious diseases, but to research in general. So I think it's important to understand when you're sharing data, at least, in the very least, you share the method, and not just the typical summary of method you get in a paper, but in a manner that people can actually read and reproduce the exact experiment, if not the results, because obviously you can't guarantee that. So that's something we take very seriously when we provide methods material, not just in our papers, but on our website. And the other thing is also what is a hit? That there are myriad definitions for hits, so I think criteria should be well defined. So sometimes when you work with a PDP, like MMV, they have very rigorous definition for what a hit is, what a lead is. So when we work with them, then we know what they're asking for. We understand why they're not interested in a particular project, because it's spelled out right there. You have to have so much potency and so forth. But when you read a paper, whether it's in Nature or Science, you look at the abstract, new compound, new antimalarial compound discovered, and you compare all of them. There's such a huge range of potency. Novelty sometimes is not always a good thing when you don't read the fine print.
Excellent. And David, so I was curious. So with for-profits, I can imagine there's got to be a little less open access. And so, how can AstraZeneca participate in some of these other open access initiatives?
So I think there is a place for open data, and there's a place for open innovation where the data is not so on show. So we routinely share 250,000 compounds with different groups engaged in not-for-profit research. And if I was to try and get agreement from the AstraZeneca chemistry community to release all 250,000 structures, I know what the answer would be. And also, there's no value in releasing that data in the first instance. Nobody's going to comb their way through it. So it's not openness for openness' sake. One thing that we've become very aware of is that when early researchers are developing assays, actually working out how robust the assay is it going to just be plagued with frequent hitters, compounds with bad physical chemical properties? This is an issue that really arises quite early in assay development. We are shortly going to be putting out a set of 600 to 800 structures, which we are actually saying these compounds are bad actors. Our experience internally is that these are protein killers in assays. So if you develop an assay and your assay just falls over when all of these compounds are screened, you've got a very, very finicky assay. Actually, you may want to reformat your assay so that it's a bit more robust, and you're just not going to be on such a knife edge. And we see real value in releasing that data. And so I think our stance is we'll release data where we think it will have a genuine impact, not just because it's perceived to be the okay thing to do. I think there has to be a scientific rationale. If there is, I'm having a great time trying to persuade my colleagues to release 800 structures all at once, but I think it's worth it.
Excellent, great. And I know we're running short on time, and I want to allow the audience to ask some questions. But the last question I had was, where do you see open access going? I think we've touched upon that a little bit, but 5 years, 10 years from now, we have change. Everything's changing with Twitter and Facebook. So, where do you see open access for research, specifically NTDs, going? We'll start with you.
So I truly hope that it becomes more and more open access, like DSC does. But I think for the European lead factor, as we see it working together with 7 pharmaceutical companies, but also the academics are quite protective of their IP, or the technical transfer officers of the academia. That I don't think we can fully open up everything, like David was saying. I think there will be a future for sharing more amongst each others with some openness, but not complete openness. But I like the model where we can share more and more and be open more, and like you were saying, Raymond, and not repeat each other's mistakes. And I think this is a huge invest of pharma company goes into running programs that probably have been run by a different company, which have failed, but nobody has talked about it. So I think that's a shame. So yeah, I think we would benefit from more openness, but to a certain extent.
Yeah, I wonder if you could almost create a negative results database where, I'm assuming no one's going to make any money off of that, but that really stops that problem, and you're reducing all of that.
You're going to save money. That's
Exactly,
also,
exactly, yeah.
One thing I would really like to see is I'd like to see the research funders do more to mandate open data. Actually, in the UK, a lot of public money goes into research, and not all of that research sees the light of day. I was involved in a Royal Society of Chemistry exercise where we picked 750 chemistry PhD theses at random, and they contained 50,000 organic molecules which are potentially of some use to somebody. And when we actually looked against the public databases, what was actually searchable and retrievable, slightly less than half were in the public domain, and yet we deliberately focused on 750 theses that had been bought and paid for with public money, and half of that output wasn't out there in open data. And so I feel that the funders could do a lot more. Actually, we're not helping ourselves to have open data because we're not putting that front and centre in what we ask researchers to deliver.
Excellent. And Paul?
I think that's a great idea, and I think to build on that, we need ways where people can get the data out where it's perhaps not of sufficient quality to get into a high impact journal. Perhaps the experiments failed. So we need to find ways of letting groups get that data out in an easy way. And I think we're going to need ways to actually handle all that data that's coming out, and that's going to be a real challenge going forward. How are we going to build databases and tools that don't just drown us in data, that we can have the maximum impact.
Would you like to?
So yeah, I'd like to just echo a couple of points. I think someone this morning already mentioned it's very expensive to publish even one paper as open access. So if you are a prolific researcher, there's a problem there. A second is, I think, Paul just alluded to this, is we heard about the passport system, we heard about CDD. I think we need some sort of cross-platform compatibility so that I don't have to talk to you, and then talk to 5 other people. And then there's, PubChem is in there somewhere as well. So I think, again, that's what Paul alluded to as well. And finally, just to echo, I think it's so important to find a way to give people incentive to publish failures. I think if you look at literature, so there have been so many high impact paper or even middle impact paper saying, this is a great target, this is a great lead, and then that's the last you ever hear of it. And then I have actually called up some PIs and professors and asked what happened. Oh, the person graduated, funding stopped. Well, that's actually important to find out. Now, obviously, you don't write a paper saying so-and-so graduated, so we need to find a way to make that actually part of the database so that you don't, in some cases, they found out the compound was toxic, but no one will publish a paper saying, my last paper was not as important as you thought. So.
Excellent. Yeah, thank you all so much. It was a pleasure.